you have listened to almost 10 fantastic lectures, and you probably want to go home. And you probably have a good dinner waiting for you. But before you do that, I can guarantee you we have 3 fantastic lectures coming up, addressing 3 important questions. First is, what is the ethic behind genomic testing in children or newborn? Second is what's happening in the Hong Kong Genome Institute. What have they done in the past 5 years that worth our cheering for? Last but not the least is that with the genomic information and potential collaboration with Greater Bay Area, what is the drug development going to be like? So our next 3 speakers is going to help us to look into these 3 important questions. Our first speaker is Professor Bartha Knoppers. She's actually a fellow Canadian. Actually, I'm from Canada originally as well, but she's from the French side of things, from the McGill University. She's actually the one who holds multiple degrees, both actually so-called recognition in medicine and law. So she is actually a professor emeritus in the McGill University, and also she's actually the chair of the cancer research in law and medicine from 2001 and 2024. That's 23 years in the post. And she's actually also the founding director of the Center for Genomic and Policy of the Faculty of Medicine and Health Sciences at McGill University. So I could not find a better person to address this important question. What is the ethic behind genomic testing? Thank you.
Good afternoon, everyone. Bonjour, as we would say in Quebec. It's a pleasure to be back here in Hong Kong, which actually is my favorite city in the world. This lecture is so completely different from everything that you've heard today, so hopefully you'll find that it is an introduction to the real world behind the statistical AI and rare diseases in terms of the genomics and genetics. This is my first introduction 40 years ago, exactly, to newborn screening. I was a law professor first year, and I received a call from Dr. Scriver, Charles Scriver, a famous Canadian pediatrician, and he said, BMK, I need you to go to the United States for a conference on liability in newborn screening. And I said, Charles, what is newborn screening? So that's 40 years ago, and since that time, left pretty well newborn screening for 30 years and moved on to population biobanking and all kinds of interesting developments, because newborn screening, biochemical-based newborn screening, was the most successful population public health program in the history of WHO, and it was rolling merrily along till we came not only to genetics and genomics, on which obviously it's also based, but to whole genome sequencing. And that, in relationship with the Genetic Alliance, with ERDIC, and more recently with the Lancet Commission on Rare Diseases, brings us to this issue of numbers, when, where, and should we or not involve genomic whole genome sequencing in the newborn period. I don't think we have much choice. It's actually children that are the immediate focus of genetic diseases. So we need to look at what are their rights, what are their best interests, and what do we need to do or not need to do. Now, there is a numbers working group who probably, in the Lancet Commission, will probably contest all this, but the figures are there and they speak for themselves, and the period of time is short, and our focus has to be on what is best for the newborn. These are some of the figures that you see in terms of how not only the number of years of diagnostic odyssey would be reduced, the results would come sooner, and hopefully the actionability, not just treatment, but actionability surrounding the newborn and their family would begin much earlier with changes in outcome for the child and the family. About 3 weeks ago, I went to the second, or maybe more than that, meeting of ICONS, the International Consortium on Newborn Screening. And this consortium actually has expanded to twice the number of research projects that you see on the screen. And more recently, it is definitely international. There's a great interest in most countries in incorporating whole genome sequencing into their newborn screening program. So what I'd like to do is give you a brief introduction, talk about the health rights of the newborn, discuss whether maybe yes or no genomic newborn screening is an option that countries should seriously consider and perhaps even implement, and then look at an aspect that has not been discussed until about 3 weeks ago or so at that ICONS meeting. So the Convention on the Rights of the Child, each of these international conventions, treaties that are formally, legally adopted and ratified, signed and ratified by countries, create actionable rights on behalf of the persons involved. So this one, the Convention on the Rights of the Child, was adopted in 1989. It has the largest number of signatories and ratifications in the history of UN legal treaties. So it's a legal treaty that can be used as a basis for asking governments, what have you done with these rights? So 2 articles or 3 articles from the convention, the best interests of the child are primary. It doesn't matter what you think as a parent, or the best interests of the child are primary. Most importantly for those here present today, the highest attainable, remember it says attainable because obviously it's contextual, standard of health, access to facilities for treatment, all are part of this definitive right of the newborn. Equally, there's the right to science. This is what we call a Cinderella right. It's a right that's been in different declarations and so on since 1948, the Universal Declaration on Human Rights, but it's being completely neglected. Yet children as citizens, as legal persons, also have the right to enjoy the benefit of scientific progress and its applications. More recently, I've begun research into if we do include whole genomic sequences in the newborn screening arena, what does that do, or how do we frame it, or how do we protect the privacy rights of the child? Now, the Article 16 of the Convention doesn't really give much attention to the data aspects, if you like, or the privacy rights of the child. It's in a general article. So what does this mean? Article 24 is probably one of the only articles in treaties and conventions that, beyond reference to children and their families, or disabled persons living with disabilities and their families, actually includes within the Article 24 on the rights of the child to the highest attainable standard of health, to ensure appropriate prenatal and postnatal health care for mothers. So you have a right that has another right for another legal person in this convention. It's very important because we're slowly moving into prenatal therapies, and this will definitely be secured by having such appropriate prenatal and postnatal included within genome sequencing programs. Now, we all know that sequencing is not treatment. Sequencing is detection. It's looking for, it's finding. It's not treatment, it's not a diagnosis. This is very important because everything else, once that at-risk asymptomatic newborn is found, all the other rights of everyone, the state, the parent, the family, and so on, come into force. But till you find the at-risk asymptomatic newborn, you don't have confirmatory or other forms of diagnosis. So you're looking for the at-risk newborn. Now, many states, unbeknownst to most persons, have obligatory newborn screening as it currently exists, the biochemical classical newborn screening, and with no questions asked, is considered part of classical pediatric standard of care. So there's 13 European countries and many American states and so on that consider it routine to do the heel prick blood test on filter paper on newborns. So if they are considering adding other kinds of tests, they usually give out a separate and full sort of consent to anything additional to what is routine. The rest is being inferred or implied that the parents are consenting. Sometimes there are religious objections. I would also consider that the failure to detect, the failure to find this at-risk child, for me at least as a child's rights advocate, is or could be considered medical neglect. Medical neglect has been used in cases, for instance, where because of religious beliefs, minor children who are not capable of consenting on their own were being refused surgery, life-saving surgery in the case of, let's say, Jehovah's Witnesses, and the state would overrule the parents' decision, saying that it constituted medical neglect. And the state, in fact, does have duties both to protect children as a vulnerable population, whether their parents do it or not, or whether programs exist or not. This is a state obligation. And finally, I would argue in this sort of visit of children's rights that children also have a right to research. And that's where that right to science, to benefit from scientific progress comes in. So these are rights that most people don't know exist. What's going to happen under genomic newborn screening? Well, you don't want to destroy or affect or change the most successful public health / population program in the WHO history. So how are we going to, if we do introduce it, not affect the classical, or perhaps keep the classical, but then what do we do with the whole genome sequencing? And there's also issues when you're doing a whole population. There's increase in false positives. There's obviously, you have to look at cost effectiveness, sensitivity, specificity, and so on. The numbers are very important in order to be able to stratify and understand what's happening in the newborn population. So these are some of the projects. Like I said, we're up to about 70 of them. They're all research projects, though. So everything that's happening, all the knowledge coming out of these research projects are based on research consents, research ethics. They're not based on public health programs or population public health initiatives. And they cannot and would not be reproducible in the population sense of having 20 pages of a very informed consent with all kinds of tools and communication back and forth and so on. Currently, in these projects on whole genome sequencing, they all obtain explicit parental consent for residual blood spots or for a new sample. Most of them, as is usually the case, obtain this verbal consent or written in the case of whole genome during the prenatal, and it involves a number of health professionals. Again, is this translatable into a public health program for all newborns in any country at any time? And interesting, this is an older slide of 2023, but it was confirmed about 3 weeks ago. In all these projects, there's only actually about 55 genes that they're all screening for and that are in common, which shows you that this notion of whole genome sequencing and screening hasn't really integrated into the science yet of that particular time period. So on this second part, on the health rights, there was an article by Jan Friedman who, in the challenges, he says, what are we going to do about facilitating informed consent, implied or consent with, or notification with opt-out or whatever, without impacting uptake, because uptake is usually at 99%. How can we do this at low cost, high speed? What about interpretation, penetrance? What happens if the variant classification changes from benign to pathogenic, from unknown to known, and so on? Imagine the workforce that is required when you start, even if you use panels, And currently, all these research projects are only screening for between 200 and 500 conditions. So they actually create a panel to in order to only sequence for those conditions. What about the impact of uncertain results, equity of access, and finally, the last one on integration of genomic data with medical records, enabling reuse. And that brings me to the one challenge that I've been looking at consents in whole genome sequencing in newborns for about 4 years now. Then suddenly I thought, well, wait a minute, if you do this and you have it, what do you do afterwards? I mean, in the classical biochemical, they either keep the cards with the blood spots for a certain number of years, just for quality assurance, for confirmatory testing and comparison and so on. But most states don't want to keep those cards in their basement lockers or otherwise. But with the data coming from the whole genome sequencing, even if you do have panels, you do have the sequence of every newborn in your possession. So we have emerging new obligations. How long do you keep them if you keep the data? Can you reuse after a certain period of time? Can you revisit for classification validation? Do you need to recontact the parents? Do the parents expect that because you've got the spots, you will be looking after their child? You will be telling them if there's something new has come up or that you've changed your mind about what you said the child had or not. And finally, with that amount of data, is there a danger of re-identification for other purposes like forensic or so on? Now, no attention or very little has been paid, and I've just started looking for privacy codes. What about children, and what about when they become capable minors? And they have to decide for themselves, do you recontact them? And there's very little except in the digital world, you know, the world of what they're allowed to see in the social media and so on. Norms are beginning to emerge, but not the kind of norms that are specific to health-related interventions or screening or research or testing. So this is a very nebulous field. In fact, the 5 R's or 6, whatever they were, scares me more than the consent issue, where I think we'll gradually find a modus operandi particular to each culture in terms of what they think they need to do. So in conclusion, I would say that the detection, the finding, is the right of the asymptomatic at-risk child. I don't care what religion, what culture, personal anti-tech or pro-tech or whatever parents or potential parents are, this is the child's right to be found. After that, all the norms, particular to different countries, can come into effect. Even for adults, we're beginning to ask ourselves, with all this genomics and genetics and sequencing, can't really do the whole clinical informed consent in detail route. We can't overload parents during the prenatal or at birth period with all this data. Can we find, not only for newborn screening, but generally in society, a different approach, what we would call more an appropriate consent, instead of the fully informed, explicit research consent that's often the ideal? And this is filtering through for adults already in terms of understanding and using genetics and genomics in research and in clinical practice. And in Quebec, as you can see on the right, we have currently in the classical newborn screening, we simply say parent was informed, yes, check it off, it goes in the medical record, good thing to have there, and parent refused. If there is a parent that refuses, then parent has to sign and the name has to be on there. So we have more of a notification with opt-out, which might be the right thing for population studies and population sequencing. So are we going to move towards appropriate consent generally, and more specifically in the newborn context? What would be the form that would be appropriate? Take the classical and say this is it, it's happening for everyone, and then have another process, more explicit, more verbal, more intervention for consent to genomic. The projects that are ongoing right now have used such, I don't know how applicable or how the interpretation of such a research consent model will work in population studies where every newborn will be screened. So I would argue that newborns are not patients. They're citizens, they have their own legal rights, and the right of the asymptomatic but at-risk child comes first. So these are some future perspectives or systemic issues. This is big stuff. I mean, newborn screening has been really safe. It's a beautiful program. And how are we going to integrate this new knowledge so we can find those children, increasing number of children in the early period when we can do the most and when they need to be found, not when it's too late. Discussions are starting. This is a discussion in Canada that was just on the classical kind of screening by the Canada's Drug Agency, where they put the health rights of the newborn as the filter for future programs. They didn't pronounce on newborn screening, genomic newborn screening, but they said, let's start, if we are going to go there, let's first start with the health rights. And then this just came out in May from the National Academy in the United States. Again, only on the classical kind of screening, but they showed that the systemic issues are as important as the ethical issues. If you're screening using whole genome sequencing, it has to be for treatable, actionable in the first 5 years, and otherwise the whole system meant to benefit the child will not come into being. So in conclusion then, we're going to see it is coming, but I think it's up for us to start the public conversation on how best to protect the highest attainable standard of health for all newborns. Thank you.
And let's see if our crowd have any questions for Professor Knoppers. Please do raise your hand if you do. Any questions or comments for her?
Hi, thank you very much for your presentation. Helen Malherbe from South Africa. A bit of a curveball for you. I just want to ask about progressive realization. This is something that's in a severely resource-constrained environment, such as South Africa, where we have many competing health priorities, most notably HIV/AIDS. We have, it's such a constrained environment that progressive realization is often put as a barrier for not implementing services. We don't have even what you call, traditional newborn screening in place. Less than 0.1% of our annual births undergo biochemical screening, which is probably shocking to a lot of you, but we are trying to get that in place. So my question is, any silver bullet to overcome progressive realization as a hurdle? And how can we also stop the gap getting wider between countries such as Canada and other highly developed countries getting further and further ahead as we get further and further left behind? Thanks.
Thank you. I'll start with the last end of your question. The traditional classical biochemical screening is used in quite a number of countries, not because they can offer a PKU diet or that there's treatment for the 3 or 4 rare conditions that they do screen for, which even Quebec, for instance, we only have 6 conditions. The screening is used as a way of making sure that newborns are registered. Because it's a public health program, the public health nurses who will go around the country to find the children and register them and do the heel prick test, it's actually a way of seeing that a child exists. So it has benefits beyond the particular diseases which may or may not be screened for at all in different countries. And so the obligatory nature of the public health populational approach ends up benefiting children as a population, whether or not the country can afford the treatment, even under classical screening. So I don't think the goal is to rush this whole genome, even with panels and so many conditions. We haven't figured out at all what we're going to do with the data and the five R's or 6 R's that I had up there. I do think we need to consider, however, that if in that early period we can immediately find asymptomatic newborns at risk, that it's our duty to protect those newborns. However, a state decides to introduce it, like probably slowly, very slowly, till they need to have their guidelines, their governance, and all of that in place.
Thank you. We have time for one more question. There.
Thank you so much for your talk, Bartha. I'm Dr. Lucy Mackay from Medics for Rare Disease, and I'm on the commission. Those 6 R's keep me up at night. And I'd say I'm from the UK, and I would say that the generation study in the UK, in England, is contentious.
It's been what, sorry?
It has been contentious and not widely accepted. And one of my concerns is when you look at all of those research projects, they're very different. You've even seen it in the conditions there, that only 50 overlap genes, many of which could potentially be on the biochemical test screening, maybe. But in the UK, we only do biochemical testing screening for 9, but we'll take the genomes for 200 conditions. And so these studies, when they're put on a screen like that, might seem very similar, but they're not. And I think one of my questions here is, this is research being done in children who can't consent, and they really should individually have a benefit. It shouldn't be a group benefit. In the UK, this is meant to be a 1% pickup rate. What about the other 99% of babies who are going to get no benefit themselves from this research, but are being put through it? Their families are going to have to be reconsented. They have all of this admin. Their data is, they haven't decided about the 6 R's. And I'm genuinely asking, is it ethical for 99% of these participants to be in these studies if they're not going to have a direct benefit?
Well, as a Lancet commissioner on rare diseases, if we start playing numbers like that, we're not going to get very far, period, in terms of the science of rare diseases, to say 1 is too little, 3 is too much, and so on. I think the point is, if the quality of the science, the integrity of the science, and the treatability of the conditions in the first 5 years of life is shown to be of benefit to those at-risk asymptomatic children, and it will take time for those research projects to show that kind of data. So on that score, you're right. I don't think we can, in advance, start sticking numbers on what counts in terms of finding these children.
Thank you so much. Thank you for the questions, and thank you, Professor Knoppers, for your time. And now I'd like to invite Professor Tony Mok once again back to the stage and introduce our next speaker for us.
Our second speaker, Dr. Brian Jones. Now, Dr. Brian Jones is an clinical associate professor from the Department of Pediatrics in the Hong Kong University. Now, his expertise is obviously on medical genetics, and he actually earned that degree from University of Toronto. So another Canadian approach to the situation. And since then, she had come back to Hong Kong and won many awards. And currently, he's the Chief Medical and Science Officer to the Hong Kong Genome Project, and he's going to tell you everything about it in the past 5 years. Thank you so much.
Thank you very much. This is a special presentation for me. It's special, first of all, because I have Professor Mok giving that very kind introduction. And I think the second thing that is special is usually at this time I would be saying something like, thank you very much for the organizing committee to invite me. And this time I cannot say that. But instead, I really want to thank the audience from local, from China, as well as from international. What you can see here is I didn't put my name here on the title slides because this really isn't my work. This is the concerted efforts of a lot of people. It takes a village of people to do the work that is done in the Hong Kong Genome Project. First of all, the generous support from the Health Bureau, our board, our chairman is here, the advisory committees, the collaborators, the clinical collaborators, the scientific collaborators, and the advisors, and of course, colleagues and staff in the Genome Institute. So this is a presentation that I would like to first start with the acknowledgement. So genomics is changing the clinical paradigms from diagnosis to detection to treatment. And it's quite a common practice internationally to have governments and countries actually using Genome Project to accelerate the adoption of genomic medicines in their own places. So the Hong Kong government is very forward-looking. Back in the year 2017, the Chief Executive Policy Address actually has suggested a setting up of a steering committee to lead the development of genomic medicine in Hong Kong to tackle all these challenges that is listed here. So the Health Bureau is the overall coordinator of the Hong Kong Genome Project, and the Hong Kong Genome Institute was set up in year 2021 to execute this project. From the very beginning, we understand the importance of collaborations with stakeholders, so we collaborate with Department of Health, the Hospital Authority, and the 2 medical schools in Hong Kong. I'm very glad to let you know, up till now, we are already able to recruit patients and participants from 12 hospitals under the Hospital Authority, and I think I owe a big thank you to many of you sitting here. So our vision is actually to avail genomic medicine to all for better health and well-being. We do this by integrating genomics into medicine. So I want to let you know that the project actually started in the year 2021, and the 5-year goal is actually to recruit 50,000 individuals at our 5th year. 2025 actually marks the 4th year of our project, and I'm happy to let you know that we have already recruited over 52,000 individuals into the Hong Kong Genome Project. So we have 3 main themes: undiagnosed disease, which is very related to rare disease in Hong Kong, hereditary cancer, and of course, genomics and precision health. So informing and engaging the public is very important. Just now we have some discussion about newborn genome screening, and therefore we actually cares a lot about informing the public in Hong Kong about our project and at large the development of genomics medicine in the whole world and how genomics can serve the local population. And I'm very glad that we have a lot of attention from the media, and there is a recent South China Morning Post editorial that actually mentioned our project as an evidence of our progress on biomedical hub development. I'm very pleased to let you know that we have established Hong Kong's first end-to-end whole genome sequencing infrastructure meeting international standards. And our work didn't just stop here. And I just want to highlight a few developments in the past 1 year about how we refine this infrastructure. We have developed an integrated analysis platform to do our genome analysis. We have to make our own platform because many of these commercial platforms that is available could not serve all the purpose. So this particular platform that we have developed, called Cura, supports the analysis of both short-read and long-read sequencing, and it supports the analysis of different types of variants. And it also allowed us to use directly some of these guidelines, for example, the one by ACMG for variant classification. What it enables us to do is also a learnable healthcare system. So every time when we see a new patient and analyze the genome data, we would be able to know from the support of the Hong Kong Genome Project knowledge base whether this is a patient that has variants that has been seen previously in the Hong Kong Genome Project, and therefore it facilitates and accelerates the process of genome curation. And at the same time, information can go back into the system so that next time when we see the same variant, we would be able to realize that as well. And of course, clinical information is very important, and this platform also allows us to use tools for variant prioritizations using clinical information. For example, exomices that has been widely used in the UK 100K project, and also new versions that we call genomices that allowed us actually to extend our prioritizations work into the non-coding part of the genome. So part of our work of optimizing Genomizer into our workflow has been published earlier in the briefings in bioinformatics, and we have proven that the workflow that we have developed is also capable of detecting and identifying technically challenging variants that Professor Kim Boycott has mentioned earlier. So we were able to see that actually up to 20% of all those variants referred to the Hong Kong Genome Project is belonging to these difficult to sequence variants, including variants in the intronic regions, repeat expansion, structural variants, and so on. So we also look into how we can improve the efficiency of our analysis. So typically, analysis of one whole genome data set takes around 3 hours. So earlier this year, we tried to see if we can accelerate the process by developing a bespoke semi-automated analysis workflow. So this is developed by the bioinformatics team of our institute, and we're able to group patients with similar clinical characteristics into one cohort and exercise this semi-automated analysis. What is the results that we have achieved? We have been able to shorten the analysis time from 3 hours per patient to around 20 minutes. And we have already used this platform to do a field cohort analysis, including those that are listed here, with the capability to attain a diagnostic yield compared to international standard. So working with colleagues, ophthalmologists from Grantham hospitals, we actually have published one paper that used this semi-automated cohort analysis workflow for patients with retinitis pigmentosa with some success. Now you notice the word that I use is semi-automated. We cannot do full automation because a bottleneck that we can see in our analysis is how do we read information and data from medical literature and use those to support evidence that is needed from reading the medical literature. So earlier this year, all of you would hear from news about the development of a new LLM model called DeepSeek R1. This come to our observation because we're able to make good use of the very important characteristics of DeepSeek R1 being very transparent in the decision-making process to help us to control AI hallucination in reading the medical literature. You can see here that we do some benchmark using human genome analysts as a comparison, and we were able to show in our hands that we are able to see a very high concordance rates in the results of the analysis, being 96% in the primary analysis and 98.7% in the reanalysis. And currently, we are now adopting the DeepSeek workflow into our genome analysis so that we can help our genome analysts to further improve the efficiency of genome analysis. So these are all the works that we have done to set up a good infrastructure. And the purpose of us working in the Hong Kong Genome Institute to set up good infrastructure, good workflow, is really to enable good collaborative projects with our colleagues around Hong Kong to serve the Hong Kong population. So I'm going to give you examples of 3 studies of good collaborations, and it is really through these collaborations we can demonstrate the power of genome sequencing in healthcare. So for the first studies that I'm going to talk about is on our pilot phase studies of our undiagnosed disease project. So you see here our collaborators led by our clinical leads, including Professor C.S. Lau, who has just been our moderator for the AI sessions, Dr. Jackie Lam from CUHK, and Dr. Luke Ho-Ming from the Children's Hospital. So on this pilot work, we used the combinations of short read and long read genome sequencing, and we were able to reach an overall diagnostic yield of around 28%. Interestingly, 1/3 of all the variants are novel. That has not been reported in medical literature, showing the importance of all of us have to do our own homework when we are implementing genomic medicine into the local practice. These are potentially variants that are more common in Hong Kong compared to the other place. What does it help? What does it help for the patients? We see that we are able to end very long diagnostic odysseys in our participants with an average of 15 years. So for those, we can get a positive diagnosis in 77% of them, that it actually provide a positive impact in the clinical care of these patients. And it is very meaningful because this paper has been accepted for publication in the Lancet Regional Health Western Pacific in February, which is the month we celebrate rare diseases. So I want to give this second example, and this is on our cohort on hereditary cancer, and this is a collaboration with the team from Chinese University of Hong Kong and Prince of Wales Hospital, and this effort was led by Professor Tony Mok and his team. So in this group of around 1,000 individuals, we actually offer whole genome sequencing, and we do not use the stringent classical clinical referral criteria, but we use the criteria of a personal history of cancers. So what we're able to see, so let me go back, sorry. What we are able to see here is in 6% of these 1,000 individuals, we can identify hereditary cancer predispositions, which is a very important information. So compared to the usual clinical referral guidelines, what our approach can act is a 30% more of capability to identify patients with a familial implications. So this is, of course, very important. Firstly, this information would be very helpful to refine the individual cancer surveillance of these patients. So there are already established international guidelines for genes like BRCA1, BRCA2, Lynch syndrome, to help us to improve the clinical care of the patient. This is very helpful for the family because the genetic test would be able to provide an opportunity to do effective family screening for these cancer conditions. And of course, nowadays we talk about targeted therapies. And many of these important therapies like PARP inhibitors or immunotherapy, now the inclusion of genetics information would be a reason why we can make these patients eligible to be included in the treatment plan using these new treatments. So this is very useful. So our third themes, as I have told you, is on genomics and precision health. And so far, we have already more than 20 different cohorts recruited into our project. You have heard Dr. Derek Lee, Dr. Becky Ma talking about our cardiovascular cohort and renal cohort. And after me, you will hear from Aya to talk about our work in brain cancers and how does it facilitate clinical trial. For me, I would take this opportunity to introduce a collaboration between us and the 1997 birth cohort team of the School of Public Health of the University of Hong Kong. So this is a very famous birth cohort in Hong Kong. And for those of you who do not know about this cohort, this is a cohort on patients that were born, on children that were born in the year 1997. And it has already produced a lot of good research results and make us understand the early life determinants of long-term health outcomes in Hong Kong. So what this project has also done is the collection of biosamples, including DNA, and importantly, at 17 years of age, these participants has been called back to safe sample for metabolomics studies. And as you know, metabolomics are important snapshots of an individual's health status and disease risk, and is now being used as biomarkers for disease predictions and monitoring. So what we have done in these collaborations is based on two statistical genomics methodology. First is GWAS, Genome-Wide Association Study, to see if there is new discovery. And we also employed another statistical genomics method called Mendelian randomizations to study the associations of the metabolites and some important disease outcome. So first of all, let me report to you our findings in the GWAS studies. So typically in GWAS results, you would see Manhattan Port. This is a representation of a compressed version of 250 Manhattan ports, because in metabolomic studies that we have done, we have included 250 different metabolites and metabolites ratio, and what we are able to find are 55 independent associated locus, with 9 that is novel and only found in this Hong Kong cohort of patients. So let me show you one of these important findings. So this is a non-coding gene. The name is over there. And the significant SNPs is at the transcription start sites of the gene, and it is associated with extra small VLDL metabolites. What you can observe here is a clear dose-response relationship, and I want you to note the reason why this is novel finding and we are able to find this is because of the very different in allelic frequencies seen in the East Asian as well as in European population, meaning that we are finding some novel finding that are directly applicable and important to the local population. Well then, does it correlate with future outcomes? So we are dealing with aging populations. There are a lot of cardiovascular disease occurring at the later stage of life that we want effective way of prediction. However, 1997 birth cohort is a young cohort. All of these participants are younger than 30 years of age. So we employed this methodology called two-sample Mendelian randomizations. So we make use of the GWAS of the 1997 birth cohort and the other published outcome GWAS, including stroke, hypertension, heart diseases, and we focused on those significant SNPs that overlaps these GWAS studies, and using two-sample Mendelian randomizations, we are able to correlate and see if there is any association between the metabolites and these important outcomes. And long and behold, we are able to see that several important metabolites taken at the age of 17 in the adolescence period are really useful to predict some adult onset outcomes, including these conditions that I have listed here. And I want to point out to you stroke. In terms of stroke, it is that novel marker that I have introduced to you in the earlier slides that actually shows an increase in the risk of stroke there. So now we are using genomics data not only in diagnosis and treatment, but this work is actually possible for us to be able to do some useful disease prediction, using metabolomes as a biomarker occurring earlier in life to see if we can focus our prevention strategies on these important groups of patients who are at risk of developing cardiovascular disease later in life. So we're making good progress, and through all these important projects, we are adding in new data into the genomic database of the Hong Kong Genome Institute. And so, therefore, in the genome database, we have clinical, genomics, and sometimes even multi-omics data. We need to make good use of it to facilitate our work in Hong Kong to improve the clinical outcome and public health policies. So earlier this year, we looked into how we can better make use of our database. So a flagship initiative was initiated to see what are the additional use for those. Yesterday in the conference with the Hong Kong Academy of Medicine, I have talked a little bit about the genetic burden of familial hypercholesterolemia, and there are actually more diseases. 2 weeks ago in the presidential lecture of the Asia Pacific Society of Human Genetics region, I have shared with colleagues in Asia Pacific region that the carrier genes that is common in Hong Kong is very different from those that has been detected in Australia or in America, and people are really amazed that we really need to do our own homework before implementing some of these guidelines on screening and diagnosis. And for today, I would like to use the rest of the time to talk a bit about pharmacogenomics. We were able to do pharmacogenomics profiling on over around 10,000 individuals in the Hong Kong Genome Project database. So on this particular graph are genes that are implicated in pharmacogenomics. And the x-axis are data from the allelic frequency for CPIC reference. CPIC is an international consortium. And then on the y-axis is the allelic frequency of the same genes in the Hong Kong Genome Project. So I just want to point out, if it works, that the CPIC data are mainly about Europeans. And of course, the Hong Kong Genome Project reflects the local population. You can immediately now see from this graph that we see very different patterns. There are some genes that are more important in patients in Hong Kong compared to the European population. So what is the impact of this finding? I'm trying to impress you with two ways of representing the data. First of all, at individual level. All of us sitting here, if we're represented by the Hong Kong Genome Project participants, have at least one actionable pharmacogenomics findings, and on average, 5 of them. So common findings, including genes like VCOR, which is found in more than 90% of all our individuals. And if we are giving out warfarin at the dosage suggested for our European counterparts, then we have a risk of over-coagulation, which could be leading to dangerous, life-threatening complications. Other genes like CYP2C19 is around 40%, 50% prevalence here in Hong Kong, and it had implications in different drugs that is listed here. The other way to look into the whole this pharmacogenomics data is to use big data approach. We're very proud in Hong Kong to have a good electronic patient's records, so I'm looking only at the prescription data of HA in the year 2024. For the top 20 prescriptions, this is totaled to a prescription number of 2.9 million prescription, and out of these 20 drugs, 12 has already established evidence-based guidelines to use pharmacogenomics to guide prescription. If we correlate this with our finding from the Hong Kong Genome Project, it means that in 0.9 million of the prescription that we give with the pharmacogenomic profiling, it is actually actionable. Things can be done to reduce the side effect, to increase the efficacies of the drugs. And that's translating to 30% of prescriptions, only counting hospital authority, could actually benefit from a pharmacogenomics testing for actionable phenotypes. So this is, I think, very clear to you that both at individual level and at a population level, the importance of genomics. I'm coming to the end, 2 slides of my presentation. I just want to let you know that the government has informed us that we need to be very careful to document the outcome of the Hong Kong Genome Project. We must document in an evidence-based manner the outcome so that the project becomes sustainable. It will support the future development of genomic medicines in Hong Kong. And therefore, we cared about how our participants feel, the satisfaction rate, and we work very closely with our clinicians to collect all this important data. I just want to finish that we have already published our roadmap for the Hong Kong Genome Project to document in a multidimensional manner this clinical and non-clinical outcome, including psychosocial and economical evidence. And Dr. Annie G, sitting on the first row, is actually leading this project. And I'm also very glad to let you know that we have joined this international team to see how we can determine the value of genomics in health. It is accepted for publication in Nature Medicine, and it is going to be released on the 27th of November. Sorry. I have to show that we are among one of the institute that participate in this work. And I want to let you know that the government is very supportive. They have actually charged the institute to lay down a plan for further development in the next 5 years. And the details of this strategic plan for 2025 to 2030 is now available in our strategic plan that is available in our webpage. And with that, thank you very much for your kind attention.
Thank you so much, Dr. Brian Chong. Thank you very much for the insightful presentation. And now, let's again invite Professor Tony Mok back on stage to invite our next and last guest speaker. Thank you.
Our first speaker is Professor Aya Halali. Now, obviously, she's not a local Hong Konger. She's only been in Hong Kong for 5 years, but we in the oncology community had considered her to be one of us. So she graduated from Ireland, and also get her PhD from the Queen's University, Belfast, and also received the Sir Alan McClay Clinical Research Fellowship. Now she's in Hong Kong doing the mostly and also not exclusively on the neuro-oncology research. On the top of that, she also has extensive experience in genomic, biomarker, and also drug development in phase 1. So, yeah.
Good evening, everyone. So firstly, it's a real honor to be introduced to the stage by Professor Tony Mok. And also thank you to the Hong Kong Genome Institute for their kind invitation to present here today. on the topic of genomics as a catalyst for drug development in the Greater Bay Area. So these are my disclosures, and I serve as an advisor for the Greater Bay Area International Clinical Trials Institute. So I wanted to first start with setting the stage of what the Greater Bay Area, so I acknowledge that everyone isn't very familiar with the region. And it's essentially, it consists of around 9 cities across the Guangzhou area with 2 special administrative regions, one in Hong Kong and one in Macau. And essentially, when you look at the ecosystem in that region, which services around 84 million people, there's over 1,000 fintech companies, which are quite important when we're really talking about drug development and developing clinical nodes of excellence in drug development. But more so as well with this leverages is the over 2,000 biopharma companies that are developing and growing within this region. And this is incredibly crucial, because when we're really looking at how the infrastructure is really changing in the China Greater Bay Area, there's really a clear shift from a made in China approach or a Me Too movement to more of a created in China, with more and more Chinese biopharmaceutical companies being respected and acknowledged in the international space, particularly in oncology, as developing clear, innovative drugs that are improving patient outcomes. So there is a primary value proposition with the China Greater Bay Area for biopharma, and that's really around the infrastructures or institutions that have been established as significant and important linkers to the international and global community. And that's really with that cross-border collaboration with the 2 institutions in Hong Kong, the Chinese University of Hong Kong, as well as the University of Hong Kong. I'll go back again here. So when you see that the potential is that when you're looking at developing clinical trials and developing drugs, you have the population base to be able to test it in. You also have the manufacturing prowess, and you have the political will of wanting to change regulations that will establish more of a robust clinical service or clinical trials center. But having that idea on paper is not enough. What you need is that you need a nexus, or you need an institution that potentially will be globally competitive, but also very, very credible, because what we really need in this region is an institution that will deliver good data, good integrity, and as well as a regulation that is very well trusted. And this is precisely why Hong Kong is actually very, very important, because we leverage the international standards that we do have in Hong Kong, as well as the IP protection and the world-class ecosystem that's placed on both of the universities. But more so that Hong Kong has created a regulatory sandbox, essentially allowing for drugs that are potentially available to us from a global community to enter Hong Kong to meet the needs of patients and their families with rare diseases and rare treatment indications. Even though Hong Kong, and one of the trust factors for Hong Kong is the consistency in its regulatory pattern for drugs and medical devices, we still are in that secondary evaluation phase where we do need and do depend on the FDA, EMA, and the China FDA and NMPA for the import and approval of drugs in Hong Kong. But as of last year, there has been a shift in that policy where there is that 1+ mechanism. And essentially what that allows for is that the government had increasingly recognized that there are rare diseases, there are tumor types and conditions that actually truly need our attention very quickly, and that secondary evaluation process isn't fast enough for these patients. So that 1+ mechanism will allow for drugs that are regulated by either the FDA, EMA, or the China FDA, NMPA, plus a body of evidence from real-world evidence in Hong Kong that will allow for the approval. But not surmounting to that as well is that next year there is a plan to create Hong Kong's own regulatory body that will be at par to the FDA, CFDA, NMPA, as well as the EMA, which will again allow for drugs that we are potentially delivering and developing in Hong Kong to have harmonization of regulatory approval across all of the different regulatory bodies. And why this is important is that we're now kind of moving into and have moved into the world of genomics, which is really rewriting how we're developing drugs. And especially when we're looking at that traditional one-size-fits-all approach, which is traditionally very drug-centric, we were really relying on a statistical model that will allow us to interpret the data to give us an average of the proportion of patients that are likely going to respond. But this doesn't work. This has really failed us significantly when we're looking at disease types that are that require clinical urgency. So then we're really pushing into that world of precision oncology approach, a more patient-focused, patient-centric, and genomic-centric strategy. And this is incredibly crucial because we're now deconstructing the patient's tumor from its organ of origin to now the molecular fingerprint, whereby potentially now we're identifying new genomic alterations that we can help to feed back into that research and development ecosystem within the China Greater Bay Area. And what this hopefully will do is that we'll move away from that drug in search of a patient model to the patient in search of the right drug model, which is incredibly crucial and very important when we are talking and emphasizing a lot about rare diseases. Because as we look at that pipeline when we look at the pipeline of what precision oncology and genomic sequencing is doing, is that it is really fundamentally re-engineering the drug development pipeline, where biomarkers are no longer ancillary, but they're actually central to regulatory approval. And as you can see, the increase as well of these targeted therapies that have been approved in the setting of cancer. But more so, there's even more evidence where we're moving away from approving drugs for molecular targets based on the organ of origin to now actually moving into a more or a tumor agnostic or an organ agnostic approach. And what this will allow is that for the development, the shift in the drug development is to actually find the biomarker, develop the drug, and target the alteration pan-cancer, which we hope will be adopted to all other diseases as well. So as we're sequencing more and more tumors, what we're identifying is more and more alterations without any drug targets. So as you can see here, as we're now at 2025, going into 2026, where we're looking at these potentially tumor-agnostic biomarkers that do require our attention for even better drugs to be developed for these target patient populations. So there is a critical bottleneck here. As I mentioned, we're systematically deconstructing common cancers into a spectrum of rare molecularly defined diseases. So we're now dealing with common cancers but rare indications for drug treatment. So therefore, we need a model where we validate these molecularly guided therapies and to look for these needle in a haystack molecular targets. And what you need is that you need to have the access to the population to be able to sequence, but you actually also need to have access to manufacturing power that would potentially be able to develop the drugs for us. So when we look at the global shift essentially for precision oncology, we're seeing more and more drugs being developed. And this is demonstrated by the explosion of phase 1 clinical trials in the setting of precision oncology that are happening in the rest of the world, as well as in China. When we look at the phase 1 trial pipeline in China, this has far exceeded what the US is delivering in 2025. And there are more patents being filed in China for novel, innovative targeted therapeutics than there is in the US. So again, that gap has been narrowed. China has reached the same potential as the US. It has exceeded the EU, but it's how we can all really work together. And essentially, when we're looking at everything in a silo, the global and Chinese investigational products, I think the slides are moving very quickly. The access to the GBA patient population and the clinical network, what we do see is that Hong Kong is becoming more and more a super connector between them, where we are able then to emphasize and integrate our potential to be able to sequence the patient population in Hong Kong, but also to be able to tap into the biopharmaceutical companies in China. And where we can actually really do this is through the integration of both two important facets that are supported by the Hong Kong government: the GBA International Clinical Trials Institute, as well as the Hong Kong Genome Institute, because firstly, we need to leverage the genomic data of all of the tumor patients that we're sequencing and collaborating with in the Hong Kong Genome Institute, because this will expose a lot of potentially previously undetected, undiagnosed, or unreported genomic alterations, but more so the fact that we can actually have rapid trial recruitment, lowering the cost of clinical trials, but also emphasizing the high integrity of the data that we're reporting, as well as the operational excellence, including the attraction of both international as well as local biopharmaceutical companies. So I wanted then to say that if we have this ecosystem, this infrastructure where we can develop it, it's important to stress test it in a disease type that has been challenged, rather than using it in a straightforward disease such as potentially breast cancer or a very common colon cancer. We need to test it in a disease where we know traditional approaches have really failed us. And this is where I bring into the world of neuro-oncology, in particular emphasis into glioblastoma, where the majority of patients will pass away within 15 months of diagnosis. Traditional chemotherapy, traditional radiotherapy has not significantly changed in the last 15 to 20 years. So this becomes an important healthcare awareness point and campaign that we really need to raise. And what I wanted to bring it back is to the human aspect, to the patients that I'm seeing across the table from me. And this is a touching story of Jenna, who's allowed us to share her story. She's a beautiful 24-year-old girl taking on a very difficult finance degree. And unfortunately, 4 weeks into starting her degree, she started to develop headaches. This is Jenna looking at her CT scan, which shows a right frontal lobe tumor. Sorry. I'll just go back again. Looking at her CT scan, and sitting behind her is her neurosurgeon whom I work with, who's operating at the moment on her brain, as we see here, as an awake craniotomy. Jenna went through treatment. She survived for 3 years, and this photo was taken 3 days before she passed away. So what I want to emphasize is that no matter what we're doing, no matter how much life these young kids still want to live, we're not doing anything more to change this. And this is what really spurred on our work with collaborating with the Hong Kong Genome Institute. And what we looked at is that we wanted to sequence as many young glioblastoma patients as possible. And what we identified is a very unique change in the DNA of the tumor. In patient's tumor, and it's called ecDNA. It's double-stranded DNA that's in a circular pattern, and it's incredibly different to what we see when genes are amplified. The chromatin isn't actually compact, so it's very open. It doesn't have, it can replicate and duplicate, particularly when we're looking at EGFR amplified glioblastoma. So it actually results in a very treatment refractory disease type. So working with the Hong Kong Genome Institute, what we were able to identify is these really unique methylation patterns in patients who harbor ecDNA. We found that there's very unique patterns in the immune tumor microenvironment. And why this is important is because we're now working with the biopharmaceutical company to identify how we can actually target EGFR ecDNA in glioblastoma. But more so with that, this work has also led to the identification of extracellular and intracellular peptides through the long-read sequencing method that Brian had mentioned as well earlier, which are now developing personalized vaccines for our patients to take forward into clinical trial. So we knew this push was even more. We couldn't just stop in the genomics. So as universities across institutions, with the Chinese University of Hong Kong, particularly with the Department of Neurosurgery, in the School of Clinical Medicine at Hong Kong U, collaborating with the Hong Kong Genome Institute, with DIA, which is the largest regulatory body, and AFCR, which is a funding pipeline, is essentially pooling all of the resources that we have in Hong Kong to establish the glioblastoma pioneer, which is a first-in-kind, first-in-human surgical window of opportunity. Essentially, it's a fail-fast mechanism whereby we're not putting our glioblastoma patients onto large clinical trials and waiting for 5 to 10 years for a readout. Instead, we're actually feeding these patients into, move on to the next case, we're feeding these patients into a treatment mechanism whereby patients will be treated by a drug within a short window prior to surgery. After surgery, patients will have their tumor sequenced and as they're continuing on with treatment. And what this will allow us to see is whether or not these drugs are getting past the blood-brain barrier. If they are, we'll proceed quickly and rapidly into phase 2 clinical trial. If they're not on target, if they're not passing through the blood-brain barrier, then we actually discontinue further progression, further development of that drug target. This again allows us to fail fast with these drugs to ensure that our patients are getting access to therapies as quickly as possible. So I wanted to end on a few things that hopefully will resonate with everyone and will hopefully summarize everything that we've heard here today. Essentially, the global paradigm is set, and precision oncology, driven by the genomic data, is standard of care, and the challenge is no longer discovery, but it's in the delivery and inclusion. Apologies, the slides are just going by very, very quickly. But essentially what I wanted to emphasize is we can do all of this. We can genomically sequence. We can work with the GBA, International Clinical Trials Institute, to ensure that we are enriching the Hong Kong community and the global community to access to China-made drugs, to allow patients in the Guangzhou region to access drugs made in Beijing, Basel, and Berlin. And instead, what we want to do is make sure that there's equity here. We want to make sure that all patients get access to genomic sequencing, all patients get access to biomarker-driven clinical trials and targeted therapy. So with that, I wanted to end again with the last slide from Jenna, and instead to say that there's strength in every story and hope in every step. I'd lastly like to acknowledge everyone who's helped establish my career in Hong Kong over the past 5 years. It's been a wonderful journey, and with that, I'll stop and thank. There's my slides are kicking me off the podium.
Hopefully those are not too difficult questions, but I start with Bartha. Now, a fellow Canadian. Canadian is very cost conscious. In the screening program in general, especially in cancer, we every time have to produce a cost-effective analysis. Do you need to do that for baby screening in a way of production of a cost-effective analysis before it can be adopted by the society?
The cost-benefit analysis is the traditional way. I'm not sure that for a population public health initiative that the same should apply. I mean, the population or subpopulation in the case of breast cancer or whatever targeted is to ensure the sustainability, yes, of a universal healthcare system is very important. But with the undiagnosed children that are being missed because classical biochemical screening does not cover the population of newborns as it should, we could argue that by treating, finding these children, detecting them, diagnosing them, and treating them earlier, we will over time be able to prove the cost benefit. At the same time, if we don't ensure and protect and promote rights specific to children and their needs and their conditions which happen early in childhood, what kind of society are we?
Right. But to follow up with that, also in screening, we also try to focus at a risk population versus the low-risk population.
Correct.
Can you kind of narrow down your risk assessment for baby screening? I mean, in a way, that will save the cost.
Well, the data that I heard about 3 weeks ago at the 70 countries who participated at the ICONS meeting with their data to date in terms of the PPV, the positive predictive value. I'm surprised I've learned some of these terms. So far to date is 0.4 or 0.6 predictive value in what the first 2-3 years that they've been doing this.
I see. Okay, Brian? Obviously, there's a great success with Hong Kong Genome Project, but in the same time, UK had the 200,000 UK Genome Project. Singapore is going to screen 1/10 of the population. Abu Dhabi is going to go up to 1 million. How is Hong Kong Genome Project going to be distinguished from all the other projects? Apart from benefiting Hong Kong locally, what is the global impact of our projects?
Thank you very much, Professor Mok. Professor Mok correctly mentioned that there are a lot of ongoing genome projects. So I think it gives us great opportunities to learn from each other. So I still remember when we first started in 2021, actually we have obtained a lot of help internationally so that we do not go through the same painstaking path as the others has been doing. And those genome projects that you have mentioned, the Singapore Genome Project, the UK Genome Project, we have consulted them extensively. And that's why you can see that now we work on some projects together. For example, the last project that I have mentioned that talks about how do we determine the value of genomics. And actually, the questions you have just asked, Bartha, is about the value of genomes. And Bartha has already pointed out we can measure it economically, but we can also measure it in terms of the clinical utility, the psychosocial impact, and so on. And so these are important things that we need to share and learn from each other. And for Hong Kong, I think, for example, we embark very early on in determining that this is a clinical implementation projects. So you see that we are actually looking into a lot of important clinical problems, important in Hong Kong, but also important internationally, like glioblastoma multiforme is a great example. Thank you.
Thank you. Aya? So we've done great work in Hong Kong in the past 5 years, and I heard that your Cantonese is improving. Mamade. So my question is this. As you had mentioned that there are a number of China innovation coming up very quickly, and then a lot of pharmaceutical companies are doing extremely well in China. How do we convince them that Hong Kong is the place in the Greater Bay Area to take the leadership rather than Guangzhou or Shenzhen? You know, why would they come to Hong Kong so that we can take the lead of research project in Greater Bay Area?
Yeah, I think that that's a great question. And the point of reflection is the challenges that they're potentially facing Chinese biopharmaceutical companies are facing by running international clinical trials. They're set up to deliver clinical trials in mainland China, but the nuances of actually tapping into the international community are actually very challenging for them. And that comes from a level of the ability to communicate, but also in dealing with certain CROs that we're used to in North America or in Europe. So where we leverage our point in Hong Kong is that international hub where there is already a historical precedent set of delivering international clinical trials, either as phase 1, phase 2, or phase 3, led by physicians in Hong Kong, and we have Professor Tony Mok on the stage here, that are internationally recognized. So when we can leverage the possibility of utilizing the GBA International Clinical Trials Institute, essentially by tapping into the fact that we can access a very international patient base in Hong Kong, but also to give them also further leverage and credibility when we're talking on stage at all of our scientific conferences about Chinese-created biopharmaceutical companies. So I think it's that the leverage of the international standing that the medical community has in Hong Kong.
Thank you so much. Thank you.